Hello, we are team number 4 from the Nature of Life, and we are going to talk about viruses. The previous knowledge we have to have acquired is the discovery of this theory, who was Carl Woese in 1917. That was the year he discovered it, and he studied RNA chains. Why? Because he tried to prove that RNA chains were containing ribosomes, and thanks to this, he developed the theory of the phylogenetic theory of life, that is the scheme that contains the three structures we are going to develop through this presentation.
Divisions. Viruses are classified on the basis of their nucleic acid content, their size, the shape of the capsid, and the presence of a lipoprotein envelope. Thus, the primary division is into two classes, RNA viruses and DNA viruses. DNA viruses have DNA genomes that are replicated by either host or virally encoded DNA polymerases. RNA viruses exhibit a high mutation rate and they are very frequent in combinations, thus having a unique evolutionary capacity.
The subdivisions. The DNA viruses are divided and pox viridi, that is a double-stranded complex envelope variable. Then the herpes viridi, that is a double-stranded icosahedral envelope. And then the adeno, the papilloma, and the polyoma viridi, that are double-stranded icosahedral naked. Then are the pyroviridae and the cercoviridae. There are single-stranded icosahedral naked. Then the hyphaenaviridae. There are the partially double-stranded icosahedral envelope. Next. Then are the RNA viruses that have two branch. There are three branch that are the positive ssRNA envelope, then the with reverse transcriptase, and the positive ssRNA naked icosahedral capsid. In the second, there are the picornaviridae and the caligviridae. Then the astroviridae and the hepateviridae. And these are only these families, but then are different because have other two branches. The genome envelope have the with reverse transcripts that have the helical nucleocapsid that have the coronaviridae, and then the icosahedral nucleocapsid that have the togaviridae and the flaviviridae, with the with reverse transcriptase that have the retroviridae.
Viruses can be placed in one of these 7 classes. dsDNA viruses, single-strand DNA viruses, double-strand RNA viruses, positive single-strand RNA viruses, negative single-strand RNA viruses, single-strand RNA RT viruses, and double-strand RNA RT viruses. Double-strand DNA viruses. A DNA virus is a virus with a DNA as its genetic material and replicates using a DNA-dependent DNA polymerase. Single-strand DNA virus. The single-strand DNA virus constitutes of widespread, diverse, and important group of viruses affecting all of these domains of life. dsRNA virus. Double-stranded RNA viruses include the rotavirus, known globally as a common cause of gastroenteritis in young children.
Positive single-strand RNA viruses. These viruses are major pathogens of humans, animal instincts, and plants. Examples of these viruses are the poliovirus, which causes poliomyelitis, which is a disease that can cause people to be disabled for their whole life. Negative single-strand RNA viruses. They are usually linear, and it's common for their genome to be segmented. These kind of viruses are responsible for diseases such as influenza or Ebola. Single-strand RNA RT viruses. Single-stranded RNA viruses with a positive sense single-stranded RNA genome, but with a DNA intermediate not only in replication but also in mRNA synthesis. These double-strand DNA RIT viruses have a gap in one strand, which is repaired to create a complete double-strand DNA genome per transcription. Some examples of these viruses are herpes viruses and pox viruses.
Families. There are four virus families: Arenaviridae, Buniaviridae, Flaviviridae, and Filoviridae. First, the Arenaviridae. The Arenaviridae are a family of viruses whose members are generally associated with rodent-transmitted disease in humans. Each virus usually is associated with a particular rodent host species in which it is maintained. Arenavirus infections are some relatively common in humans in some areas of the world and can cause several illnesses. The Bunia viridae. The Bunia viridae are a very large family of single-strand enveloped RNA viruses and consist of 5 genera of viruses: Orthobunia virus, Flevovirus, Nairovirus, Antavirus, and Tospovirus. They are found in and transmitted by arthropods like mosquitoes, ticks, sandflies, and rodents, and can occasionally infect humans. Several viruses of the Bunaviridae virus family can produce mild to severe disease in humans, in animals, and sometimes in both. The Flaviviridae. The Flaviviridae are a family of positive single-stranded enveloped RNA viruses. They are found in arthropods, primarily ticks and mosquitoes, and can occasionally infect humans. Members of this family belong to a single genus, Flavivirus. Then the Filoviridae. Filoviruses belong to a virus family called Filoviridae and can cause severe hemorrhagic fever in humans and non-human primates. So far, 3 genera of this virus family have been identified: the Cueva virus, Marburg virus, and Ebola virus. 6 species of Ebola virus have been identified: Ebola virus, Sudan virus, Thai forest virus, Reston virus, and Bombaldi virus.
Virus taxonomy has made a great, tremendous progress in the last 4 years to classify today about 6,000 virus entities into more than 2,000 species, themselves classified into 287 genera and the 3 orders. The 3 orders are dsDNA, which means double-stranded DNA, ssDNA viruses, single-stranded DNA, and double-stranded RNA viruses. Next slide, please. Okay, the double-stranded DNA in a person who has a possible result on a test for an anti-nuclear antibody and has a clinical signs and symptoms that suggest lupus. Next one, please. The single-stranded DNA have vital genes encoding proteins that mediate genome replication, provide viral structure, and in some cases facilitate packaging of viral nucleic acid into the virion. And the last one, viruses include that rotaviruses, known globally as common cause of gastroenteritis in young children, and blue tongue virus, an economically significant pathogen of cattle and sheep.